This is lecture 3.2 on osmosis and diffusion. During this lecture, you're going to learn about the cell structure, specifically of the membrane, and how it regulates the movement of molecules. We're going to describe diffusion and osmosis and distinguish between them. And we are going to learn about the terms isotonic, hypotonic, and hypertonic, and how that affects plant and animal cells differently. So recalling from our previous lecture, all cells have got a cell membrane. A cell membrane is made of a phospholipid bilayer and can be described using a fluid mosaic model. Cell membranes have got selective permeability, which means that they allow some chemicals in, but not all chemicals, and they can choose what they let in or out. And the mechanism by which they're selectively permeable is really interesting. In some cases, very small chemicals can dissolve and diffuse directly across the phospholipid bilayer, but larger compounds need to be escorted across the membrane using some sort of protein. Membrane transport mechanisms are the collection of mechanisms that regulate the movement of solutes like ions or molecules through biological membranes. There are two major categories of membrane transport, depending on whether or not you need to input energy. Passive transport is the movement of molecules where there is no input of ATP or energy. And there are two primary ways in which you can get passive transport. The first is called diffusion, and the second is called facilitated diffusion. Active transport is a mechanism for deliberately moving molecules through biological membranes, and it typically requires the input of ATP. So we're going to go through each one of these in a little bit more detail. So diffusion. This is the thermal motion of all liquid or gas particles. What I like to tell students is that when you think about things as being solids or liquids, you need to consider that at the scale of a molecule, you've got these little tiny things that are wiggling and bouncing around. And the speed of their movement depends on how much energy is in that system. And our way of interpreting that is temperature. So the hotter things are, the faster they move around. The colder, the slower those molecules move. And the little vibrations or movements of each molecule are at random. They're not deliberately moving towards or away from each other. So diffusion is a process where you have the movement of molecules from a region that's got a high concentration of molecules to a region of lower concentration of molecules over time. And that movement speed depends on the temperature, the size of the particles, and how thick the fluid is that they're moving through. A horrible way of putting this is there is a reason why public restrooms are often kept very cold. And that is that the cooler the air temperature is in the restroom, the slower the movement of fecal odor particles from each toilet to other areas. And so it will keep the smell down in that area. Diffusion is something that you'll also be able to play with at home. So when you take some sugar or some salt and you put it into a cup of water, it's going to dissolve into that water, and you can have a strong solution of sugar in that water. So if you wanted to make yourself an iced tea, you might make a cup of hot tea, let it cool, and then you've got your sugar water, we call that simple syrup, and you would squirt some of that simple sugar into your glass. Now, if you didn't stir at all, initially a lot of that sugar syrup would end up just at the bottom of the glass, and there would be unsweetened tea at the top. But if you just wait, maybe an hour, over time, those sugar molecules and water molecules will all vibrate and wiggle around until they end up mixing, and there is an equivalent number of sugar molecules at the top of the glass as at the bottom of the glass. Now, diffusion across a membrane is very similar. So in this picture at the bottom, you've got a lipid bilayer that's got lots of a molecule on one side of it, the extracellular space, and very few molecules inside the membrane in the intracellular space. So according to diffusion, you should move from a region of high concentration to low concentration. So those molecules should move from the extracellular space into the intracellular space below. And they should continue to move until you have equilibrium. Equilibrium is when you have the same number of molecules on either side, and the rate at which one molecule moves from the extracellular space to the intracellular space is the same as the rate of those moving from the intracellular space to the extracellular space. All right. Now, the second mode of passive transport is called facilitated diffusion. This is when you have an escort. So you're not just diffusing straight through the plasma membrane, but you are being helped across the membrane with a protein called a channel protein. And channel proteins are specific to the compound that they are facilitating. An important category of channel proteins are called aquaporins. In their name, you have a hint as to what they do. Aqua for water and porin, hole. They are literally water holes in the cell's membrane, and they're important for helping cells to regulate water going in and out of the membrane. Osmosis is a special type of facilitated diffusion. This is something that many students confuse. If I ask you to distinguish between osmosis and diffusion, oftentimes people will get mixed up. Diffusion is the broad category that can include all sorts of things, and osmosis is a narrow version of that. It's a specific case. It is the diffusion of water, and only water, across a membrane through channel proteins called aquaporins. So you're moving water from one side of a membrane to the other through aquaporins. In biology, we have some terms referring to the strength of a solution inside of a cell versus outside of the cell. And these three terms can be sometimes a little confusing for students. I'm actually going to start with the third one, hypertonic. Have you ever met anyone who's hyper? They have a much higher level of energy than everybody else around them. Hypertonic solution is a solution that's got a high concentration of solutes outside. So if we were talking about sugar, there would be more sugar molecules outside of the cell than inside of the cell. Another way of putting it is the relative proportion of water molecules to sugar molecules is that there's more water inside the cell compared to outside of the cell. And so if water is moving along its concentration gradient from a high concentration of water to a low concentration of water, it's going to be moving from inside of the cell to outside of the cell. You can think of it as trying to reduce the amount of the sugar outside, but it can't because those sugar molecules stay there. Isotonic, iso means the same as. So if you counted the solute molecules and the water molecules, the relative proportions would be identical. So what that means is that it's pretty much an equilibrium. If water is the only thing that can move, water molecules will be moving into the cell and out of the cell at the same rate, and the proportion of sugar to water molecules will be the same inside and out of the cell. Hypotonic is lower concentration. So that's when you have fewer solutes outside of the cell than inside of the cell. if we were going to use sugar as our example, it would be a cell that's placed in plain water. There's no sugar outside. And so water is going to move down its concentration gradient from the highest concentration of water to the lowest concentration of water, which means that it's going to move inside of that cell. So the word osmotic flow simply is referring to the direction that water is going to move across that membrane. So hypotonic means that you have got low concentration of solutes outside the cell compared to inside the cell. If I just had a glass of tea with sugar in it and I said to you, is this tea hypotonic, isotonic, or hypertonic? You should stop, roll your eyes, and look at me and say, that's irrelevant, because the tonicity of a solution is a term that is relative to another solution on the opposite side of a cell membrane. So it would only matter if you were to stick your finger in that cup of tea. Then we could talk about tonicity, because then we would have the concentration of, for example, the cell inside of my finger. Now, since plants have got a cell wall, the results of being in a hypotonic solution are different than if an animal cell is in a hypotonic solution. So if a plant is in a hypotonic solution, which means that there is pure water on the outside compared to the sugary, salty inside of the cell membrane, water will move into the cell. Now, as the cell starts to fill up with water molecules, the cell membrane is a bit stretchy and it'll expand and expand and expand. But the cell wall is rigid. It is a cardboard box. It can't swell or expand. And so what will happen is that it holds the pressure of that cell membrane, and the plant will become crisp. We call that turgid. It's a special word for a plant that's hypotonic. So you want your carrots turgid, so when you bite into them, they crunch. If you put your carrot into a salty solution, what would happen very quickly is water would move from the cytoplasm in your carrot into the salty water outside of it. And the cell membrane would shrink away from the cell wall, and we would have what's called plasmolysis. And your carrot would get limp and bendy and be very flexible and soft. If you have a plant that's in an isotonic solution, there's no net movement of water particles, and the cell membrane is going to attach to the cell wall. You'll have a moderately crisp carrot, but it won't be perfect. So the hint from this is, if you ever have wilted lettuce or wilted plants that you want to eat, if you put them into ice cold water and you let them sit for a while, they will regain their crispness. But animal cells are different. We do not have a cell wall. And so if you put an animal cell into a hypotonic situation where there's pure water on the outside and water is moving into the cell will start swelling and swelling until it reaches a pressure point and it bursts, because it doesn't have that cell wall to hold it in. And we call that lysis. So if you were to inject pure distilled water into your veins, your red blood cells would explode. That's not good. Ideally, for an animal cell, you want the cell to be in an isotonic situation, and that's where you've got the same amount of sugars and salts inside the cell as out of the cell. In a hypotonic situation, where there's a lot of salt or sugar, the water would leave the cells and they would get all shriveled and grape-like. They wouldn't necessarily die, but they would lose their shape. Now, the last category of membrane transport is called active transport. There's a huge difference between active transport and passive transport. Passive transport is using the natural movement of molecules, and so you have diffusion down a gradient from high concentration to low concentration. Active transport deliberately moves molecules against their concentration gradient from low concentration to high concentration. Why would you do that? That would be something that you would want to do if there was a molecule or an atom of particular desire that the cell wants to have. It already has quite a few of those, but it wants more. And so it's going to require energy to shunt that particular desirable molecule against its concentration gradient. The classic example that's provided for most intro bio students is the sodium-potassium pump. This is a protein that uses ATP and it deliberately pumps sodium molecules out of the cell and it pumps potassium molecules into the cell because the potassium molecules are the desired ones. So for every 3 sodium that are pumped out, you can pump in 2 potassium, and it takes some energy to do that.